Thank you so much for coming today, guys. My name is Paige Simpson, as Sarah said. I had the amazing opportunity to be our seagrass research intern this summer, and I'm super excited to talk to you guys today about my research experiment. It had to do with the implications of Halodule wrightii shoot density on restorative transplant success and the presence of sediment organic matter. So to begin, I want to talk to you guys about why seagrass is important. Why are we studying seagrass here? So seagrass provides many ecosystem and economic services for us. It serves as a diverse habitat for many organisms, including a nursery habitat, so it's where lots of organisms like to come and grow up in a safe place. It improves our water quality, so it serves as a filter. It filters out those pollutants, which is super important. We also have, it serves as a wave buffer and an economic resource, so people like to fish and snorkel because of how beautiful and as much as a diverse ecosystem as it is. And it also decreases erosion, my favorite thing that it does. So it fixes that sediment with its roots and keeps that sediment from being in the water column. So because of how important seagrass is to our ecosystem, it's extremely worrying that seagrass in the IRL, the Indian River Lagoon, has decreased by 50% since 2007. So things like nutrient loading from pollution runoff, things like boat disturbances such as crop stars, shifts in environment like our water getting hotter than it should be, and even things like freshwater input can all impact seagrass health. So because this is such a catastrophic problem, Florida Oceanographic Society has started our foster program. So what we do with our foster program is we try to restore the seagrass in our area. So we do that by taking this burlap mat you see here, and we fasten about 6 seagrass shoots to our burlap mat using planting wire. So our 16 shoots of seagrass are either taken from our wet lab right out here in the Gamefish Lagoon, or we take fragments of that seagrass from, they might have been washed up on shore. We fix it to the burlap mat here, and we deploy it out into the IRL in hopes that it takes root and forms a happy ecosystem. So we use burlap mat, planting wire, and these right here are bamboo skewers to fix the mat into the sediment, all because they are biodegradable materials. So once that seagrass takes root and forms its own meadow, those materials will then disintegrate, and it will just be the seagrass left. So this summer, for our research project, we wanted to think about the ways that we can maximize how much seagrass we are restoring with the same materials and methods that we've been using with our foster program. And one way that we've thought to do this is by adding more seagrass shoots to our burlap mats. So research actually shows that a higher shoot density can indicate seagrass health. So by adding more seagrass shoots per burlap mat, transplant success can skyrocket. So it allows our seagrass to propagate and take root for long-term survival. Higher density plantings also allow for more shoots to grow and at a faster rate than with medium or lower density plantings. So with this information in mind, my hypothesis for this experiment was that by using 24 shoots per burlap mat rather than 16 shoots per burlap mat, the seagrass would have a higher chance of long-term survival once it was deployed out into the IRL. So in order to undergo this process, we had to start by making the seagrass mats. So this experiment alone required about 900 seagrass mats, which is an insane number. So here are some pictures of our amazing volunteers making our seagrass mats. Thank you so much if anybody here helped us with that process. You guys are amazing. We filled a bunch of these big tanks out by our lab. This is all seagrass mats in here, and it was quite the feat for us to get that done, so I appreciate everybody's work. So once we have those seagrass mats made, we then deploy them at our 2 sites just up north of Florida Oceanographic Society in Brevard County. So our 2 sites are Rum Cove and Hogs Cove. You can see they're right across the Indian River from each other. So what these sites look like, here's Hogs Cove. It's a fenced-in area, and we have our plots within this site. So we have 14 plots of our 16 shoot mats, and we have 14 plots of our 24 shoot mats. They're placed within this fenced area at a random order. So here are some aerial pictures that we were able to get. And here's Rum Cove. So Rum Cove is set up the same way. Here's us building the fence. And here is the random order that we have those 16 and 24. And this fence was put in place to keep any critters out that might want to use our seagrass as lunch. and that will inhibit our ability to monitor the seagrass. So after we've constructed the mats and deployed them out into the water, we then monitor them. So we monitor them at a monthly rate. So we looked at everything in June when we deployed, we looked at everything in July, and then we just took a peek at everything in August. So what do we look at when we're out there? Well, we take into account how many shoots are within that plot that we planted. We look at the percent of our shoot density, so how dense is that plot of seagrass that we planted. We look at our shoot height, so how is our seagrass growing? Is it getting shorter? And then we were also looking at expansion of grass. So are there more shoots popping up outside of the area that we originally planted? And it's always very exciting for me to see grass expansion because that means our job is doing its thing, it's working. So while we're out here monitoring our seagrass, we wanted to think of different variables that could affect seagrass health. So it's more than just seagrass when we restore. We wanted to think of a bigger picture. So the first thing that came to my mind, because I love soil, is the sediment. So sediment is key to ensuring our seagrass can be healthy and happy for a long time because of the nutrients that it provides. So regardless of whether you have 24 shoots or 16 shoots on a burlap mat, it's not going to matter if we don't have those nutrients within the soil to keep everything maintained. So the nutrients in the soil that we're looking at is organic matter. So having organic matter in the soil allows for us to have more leaf biomass and a thicker canopy structure, which is exactly what we're looking for in a healthy seagrass environment. So let's talk about this organic matter in the sediment. So how does it maintain itself within this sediment? So what happens is because the seagrass in the sediment is underwater, there's obviously low oxygen. Well, what happens with this organic matter is it stays in the sediment until it is broken down by microbes. These microbes can't break down the organic matter if they don't have oxygen. It takes them a long time without that oxygen to break the leaves down. So because they can't break these leaves down, the organic matter stays for long periods of time and allows that seagrass to get as much nutrients from it as possible. So where does this organic matter come from? Well, it comes from a varying amount of sources. So it comes from the seagrass itself, which I think is super interesting because it cycles the nutrients. So we have our above ground biomass. So let's say a leaf might pop off, it might die off. It accumulates onto the sediment, and the seagrass can get the nutrients from that leaf and continue to be healthy. It can also come from our below ground biomass. So the roots or the rhizomes might decay and would again allow for those nutrients to become available to the seagrass. We also can get organic matter from other sources. So let's say there's a mangrove leaf floating in the water. These seagrass leaves act as a filter. So that mangrove leaf will float through and the seagrass leaves will catch it and it'll fall down and accumulate into the sediment and again be available as nutrients for that seagrass to grow big and strong. So with this information in mind, when looking at the organic matter in the sediment, my hypothesis was that the sediment under the seagrass sites will accumulate more organic matter content than the sites that we tested that are under our control, which is just sand. So in order to undergo this process of my experiment, we took 5 samples for each treatment and 5 samples for our control. So we took 5 samples for our 16 shoots per mat, we took 5 samples under our 24 shoot per mat, and 5 samples for our sand control. And if you look here in this cool little soil core, you can see dark black streaks, even some shells right here, but the black streaks are what we're looking for because that is the organic matter. So it was really awesome to take these cores and to look and see with my own eyes what was available to help that seagrass grow. And this is me taking the control samples. As you can see, I'm outside of the fence, so the seagrass is inside here. I'm still close by, but I'm in an area where there's no seagrass, it's just the sand. And then these samples were taken back to our lab and tested for how much organic matter they held within them. So, what we're all waiting for, what did we see throughout the duration of the summer with our seagrass? Well, so at our first site, Rum Cove, this is what it looked like in June. It's beautiful, the seagrass is long. You can still see the bamboo shoots here, you can still see the mats. It looked awesome. We came back in July, a little worried at first because the seagrass shoots got so much shorter than how they were originally, but my team informed me that that is because of transplantation's stress. So being taken from one environment, attached to a burlap mat and put in another environment, that's stressful for the seagrass. So it's shortened in order to maintain its health, but you can still see it's green, it's luscious, there's still so much grass, so everything looked amazing on that front. And then we go back in August, and look at how beautiful it looks. The shoots have gotten taller than before. There's so many shoots around. I was seeing lots of expansion. It was awesome to see, and I was super excited. So let's talk about our other site, Hogs Cove. So again, here's what it looks like when we first deployed. Beautiful, amazing, awesome. I'm super excited going from Rum Cove to Hogs Cove. I'm on this rush of let's look at the seagrass, and I see this. So this seagrass is not looking so happy. You can see that it's about 1 centimeter tall. It's brown. The sprouts that we did see were few and far between. It was a little scary, but in my mind, when I saw this site, I was like, you know what? Maybe it'll pull through. Everything will look good next time I come. Like, it'll be okay. Well, here's what I found in August. So if you see these little dark spots right here, these are the bamboo skewers. So they're supposed to be mapped right here. And we just see nothing. But that is okay. So this graph is going to represent, again, what we saw in our seagrass survival. So these two lines represent Rum Cove, this line representing the 24 shoot mat, and this line representing the 16 shoot mat. And then we see the crashing and burning of our Hogs Cove, so we see the seagrass just taking the absolute biggest dive in July, and it continues to be dead through August. So because of this information, I just want to let you guys know that the data that we talk about from here on out is just going to have to do with Rum Cove because, well, Hogs Cove just did not give us what we needed in order to draw any actual significant conclusions. So, how did the seagrass fare? Did we see any differences between the 24 shoots and the 16 shoots in how the seagrass survived, how it grew, what it looked like? And pertaining to shoot count, we did not find any statistical significance between the way that the 24 and the 16 shoot mats grew. Additionally, with shoot density, with how dense that plot was that we looked at, we did also not find any significant difference in the way that the 16 and the 24 were able to grow. And this is also going to come as a shock, but with our shoot height, we again did not see any differences between treatments. So did sediment play a part in what we saw in our seagrass? So when testing for seagrass soil bulk density, between our 16, our 24, and our control sand, we saw no significant difference. And when we tested for our soil organic matter content, between our 16, our 24, and our control, we'll let it hang in the air for a second. I know this is suspenseful. We did not see a difference. So let's talk about it. Let's address the elephant in the room. So for the seagrass portion of this experiment, my original hypothesis, given the research that I had looked at beforehand, I thought that the seagrass that was planted with the 24 shoots per mat would be wildly more successful than that of the 16. But as we saw with those beautiful, beautiful graphs, there was no significance between the 24 and the 16 treatments. So what does that mean for us? Well, we're going to continue to monitor this project because it is still ongoing. I'm very interested to see what the seagrass will continue to look like as the months go on. And I would absolutely love to see us continue to do more research projects on what that number will look like of shoots on a map that will take us from going to the results that we have now, which are great, to even better restoration results in the future. But until we do find the perfect number that will allow us to have an increase in seagrass restoration progress, we're just going to continue using our original 16 shoots per mat until we find out what that number is. But that was super exciting that I was able to kind of start this process of figuring out how we can maximize what we're doing. So even though we didn't find anything this time, I'm super excited to see what will happen from here. So let's talk about the sediment aspect of this. So my original hypothesis, given that seagrass can contribute organic matter to the sediment, was that the sediment tested under our seagrass sites would have more organic matter than what was tested under our controlled sand. And again, what we saw was there was no difference. But this makes a lot of sense. I was not panicking when I saw this. It made total sense because soil takes a long time to form. it takes about 100 years for an inch of topsoil to form, and that's just above the water. But if we remember, under the water, soil takes a long, long, long time to form. So 3 months is just not going to do it for us, but if we were to watch the differences between organic matter in a year or maybe even 10 years, we would start to see some really awesome and interesting things, but right now, that's just not the case, unfortunately. But I'm super glad that we were able to do this portion of the experiment, because it just gives us a good scope of information to keep in mind when we restore seagrass. So again, it's not just about the seagrass, it's a bigger picture. When we put it in this new environment, we want to know what aspects to think about that can enhance the way that this seagrass is able to survive and do its thing. So I was so happy for the opportunity to look at soil. 1, because I love it, and 2, because it's so interesting and it's so important to know how it interacts with the seagrass itself. So for this project, I would really like to thank Dr. Simpson, my amazing mentor, and the rest of our amazing research team. Everybody was so supportive and just helped me so much throughout this process. I want to thank my fellow interns who were awesome. I loved getting to know them. I also want to thank our community volunteers who helped make this experiment possible. They're the ones who made all the mats, and I'm so grateful for them. It's so awesome what they did. And I want to thank the FOS staff for welcoming me with open arms. I loved getting to know everybody. Sorry. And I want to thank my family for allowing me to take this opportunity and really run with it. I would love to answer questions. Yes.
Yeah, thank you for your presentation. Having some experience in doing this, there's a variable which I think needs to be looked at more intensely, and that is the bacteria that are required to break down your organic matter.
Yeah.
And so that the grass will have something that it can absorb.
Mm-hmm.
And in all the studies that we have done here, as far as I know, we've never really looked at the biodome.
Yes.
And I think this is really an important question. So why did it grow in one area, and why did you not have success? There must be a reason, and part of it could be that the biology there is not sufficient to break down the nutrients for the grass to grow.
Yes.
So if you added that to your study, I think that would help at least tell us how important the biology is.
100%. I'm so glad that you brought that up. So as we were talking about the microbes, and as you said, those microbes within that soil, they drive so much of what that soil does for the plants that might be rooted within it. So when I landscaped about a year ago, what we were thinking about when I saw the plants might not have been living was there might not be enough microbes to support this soil system. And it's the exact same with our seagrass project. So I would absolutely love to see us do more research into the biology of our soil and what we can do to utilize that biology, possibly bring it back and plant our seagrass in places where there is a suitable microbiological ecosystem. So I'm very glad you brought that up. Thank you so much. Yes?
Why for Bar County and not Lee River or St. Lucie or Martin?
That's the million-dollar question. So it just comes down to permitting. And right now that just seemed to be a site that was most suitable for permitting matters. But hopefully we'll be able to do some more closer down here pretty soon. Yeah?
Do you have any idea the organic matter in this area is comparable to what's in the Bar County?
That's a great question. So it actually depends just even a few feet away from where you might stand. So that's what's so cool about the soil, is I could be here and the soil would look completely different than if I were here. So it probably is quite different, and that would be interesting to look into. It also depends on where it is in the water, how close the land is. Maybe there's lots of mangroves that, have their leaves out in the water, and it just depends, but it would be very interesting to see the differences, and we'll see how the seagrass reacts, and if it's healthier down here or healthier up there. Good point. Any questions? All right, well, thank you guys so much.